Right, hello. I'm going to go through some of the basic ideas about enzymes. You have learnt right at the very beginning of biology that living things have to do seven life processes. And those seven life processes, the way we remember them is by Mrs. Gren. So you've got the seven processes. So you've got movement, respiration, sensitivity. I'm going to forget them in the right order. Growth, reproduction, excretion, and nutrition. So those seven life processes, if something is living, then it has to do all seven of them. Now, in all of those reactions, enzymes are involved. So what I want you to think about is try and remember what a catalyst is and what effect it has on a chemical reaction. Okay, so a catalyst is something that speeds up chemical reactions, and that's what enzymes are doing. They're very, very special chemicals that are made from proteins, and they help to speed up the reactions that take place in our body, whether that's a breaking down reaction or whether that's a building up reaction. Okay, so enzymes are biological catalysts because they're made inside living things. So the catalyst part, it speeds up a reaction. The biological part comes from being made inside living things. Now, there's lots and lots of different types of enzyme. They all work in a very similar way, but each enzyme works on one specific reaction. Okay, so there has to be lots and lots of different types, and they each work on one very specific reaction. And the reason for that is because they all have a very particular shape. Now, hopefully you'll remember this idea of the lock and key model. Okay, so quite often, I might talk to you about, well, if I come to your house and try and get into your house with my house key, it's not going to work because locks have a very specific key that will unlock them. Okay, so I've tried to open my classroom door with my car key before and it doesn't work. Okay, so they have very, very specific shapes. So you've got there your green sort of Pac-Man. The shape where the molecule fits, the substrate fits, is called its active site. Now, if something comes along that isn't the right shape, okay, so you've got our purple substrate here, then it won't fit in. So it won't slot into that active site. You have to have the right shaped molecule to fit into the active site. Okay, so this yellow one's coming along, it fits perfectly into the active site and it forms this complex. Now, sometimes it's called the enzyme reaction complex, sometimes it's called enzyme substrate complex. Okay, but the point is that it's the two bits joined together. So you've got your substrate actually fitting into the active site, this special hole on your enzyme. Okay, now what enzymes then do is that they speed up the reaction, and they actually will split the substrate into two smaller parts. Okay, so you will end up now that our substrate molecule, that yellow one, has been split into two, and then those two products are released. The enzyme hasn't changed at all. Okay, so another piece of substrate could come along, as long as it's a yellow one, another substrate could come along, fit into the active site and be broken down in the same way. Okay, so that's the lock and key idea. So this is just in diagram form. So you've got your substrate that slots into the active site as long as it is the correct shape, it's the correct substrate. It forms this enzyme substrate complex, and then it's split into two products. It isn't always split into two products, but it's split into smaller parts. Now, if the shape is wrong and it won't fit into the active site, then nothing happens. That substrate will just float out. This one here, this diamond, needs a diamond-shaped enzyme to work on it. Okay. Now, there are other enzymes that actually build things up. So it could be that you have synthesizing enzymes. Synthesizing is to do with making things. And what happens in that case is it's virtually the same idea, but you'll have two small molecules fit into your active site and the enzyme will join them together. So a lot of enzymes, particularly those involved in digestion, will break molecules up, but some of them do actually build molecules back together. Okay, so there's some key ideas here. So we've said that they're biological catalysts that speed up chemical reactions in living things. So remember, your biological is to do with the living part, and the catalyst is to do with speeding it up. They're made of proteins, so they have a long chain, okay, of amino acids joined together, and that chain makes the very specific shape of the active site, okay. They catalyse a very specific reaction. So like we saw down here, and I don't think it'll work on this version, if it was the purple one, or was it blue, the next one that didn't fit in, if it's the wrong shape, then it will not do anything to those substrates, okay. So the purple substrate that came in, then it needs to have a purple enzyme that's the same shape that will break it down. So that's what we talk about, that's what we mean when we talk about it being specific, okay. So it can only work on one particular substrate. Now we'll come on to this again tomorrow, but at high temperatures the active site gets denatured, okay. So we use this word denatured to explain that it won't work anymore, okay. All enzymes have an optimum temperature, but again we'll come on to that in the next video. They're also affected by pH, okay, so either acid or alkaline conditions, and they all have an optimum pH as well. So they're a bit fussy, really. They like working at perfect temperature, perfect conditions. So they have an optimum temperature and an optimum pH, and if they're at those conditions, then they will work super duper. Okay. Now in digestion, there's a lot of different enzymes, but there's 3 groups or 3 families of enzymes that we need to think about, okay. And we need to know what the substrate is and what it breaks it down to. So in this case, we've got protein. So you've got a piece of chicken there. Protein is made of long chains of amino acids that are joined together, okay. And they're all slightly different amino acids. That's why you've got red, green and blue crosses there. So protease enzymes break down the protein into amino acids, so it'll break those down, so this will fit into your enzyme, and then it gets broken down into your products. They'll be broken down into individual amino acids. There are other enzymes in our body that when we've eaten chicken, it joins them back together to make something in our body. So it joins them back together to make skin or muscle, okay. So you've got a breakdown in one direction, but then you've got synthesis, you've got remaking in the other direction. So the amino acids get put back together to make parts of our body instead, okay. This is a carbohydrate enzyme. So a lot of enzymes, not all of them, but a lot of enzymes end ASE. So that's a way of remembering that they are enzymes. So this will be breaking down a piece of starch. Starch again is a long chain of glucose molecules, okay. So glucose, sorry, someone's just posted something in the letterbox and the dog's going crazy. So these hexagons represent glucose molecules, and they're all the same, okay. So starch is made up of a long chain of glucose molecules. The enzymes breaks it down into individual glucose molecules. Other enzymes might join them back together again, okay. And then the last one is lipase enzymes. So you've got a lipid molecule, which is a fat molecule, and that's broken down into 2 products, these fatty acids, the ones with the tails, and then a molecule of glycerol, okay. Now these products you need to know, okay. So you would need to, it's just knowledge, it's just that you need to learn them. So you have to know that protein gets broken down into amino acids, starch gets broken down into glucose, and lipids or fats get broken down into fatty acids and glycerol, okay. So that is a learning thing that you need to do, okay. This part, the lock and key model, you can describe in your own sort of way, but this is just learning,